Now we're going to have, our next talk is from Senjuti, who had, I think, my favorite slide from the London meeting, which I reproduced at HCA Barcelona.
Hi, I am thrilled to be here. See the smile? I am exceptionally thrilled right now after the first session because I felt the heat, I felt the air of discomfort, and I love discomfort. I live for discomfort. So I'm going to continue with that theme, and I've decided to continue making you uncomfortable. I am Senjuti. That's the right way of saying my name. I come from Bangladesh. It's a country that you think so little about, know so little about, you don't even realize that is not Bangladesh. That is Bangladesh. So in Bangladesh, I work for a research foundation called the Child Health Research Foundation. Our mission is to improve child health in Bangladesh and around the world by facilitating appropriate policy decisions through research and advocacy. Our primary work really is to provide service to patients. We are a not-for-profit organization, we're an NGO. We also conduct surveillance, and we kind of use that platform to do some research and try to influence policy changes. Why am I here? I mean, most of the scientists over here come from basic science or academic institutes. I kind of don't fit in. So, even when I was invited to the London meeting and here today, I keep on asking myself, why am I going? Well, I think for my reason, one of the reasons for being invited to this meeting again is because an event or a series of events that have occurred recently. And that includes the fact that we recently got a small-scale next generation sequencer shown over here. It's an IC100 sequencer by Illumina. But how did we get here, right? We're not an academic institute. We don't do basic science. How did we get here? Right. The story goes back to the early 1990s. And I'm going to present a lot of data, and you'll be like, but she doesn't look so old. How is she presenting data from the 1990s? I work with my father, and a lot of the data that I'll be showing you today are basically generated by his team in Bangladesh. So putting it out there. So in the early 1990s, we started a surveillance in Bangladesh, and the surveillance is basically to look for pathogens, specifically that cause pneumonia, sepsis, meningitis, and I will be talking mainly about meningitis. So since the 1990s, the surveillance has changed and evolved, but right now we conduct the surveillance in 4 different hospitals in the country. It's a semi-national surveillance, and we're supported by the WHO. And in 4 of these hospitals, we run the microbiology laboratory. So we establish the labs and we run the labs. All the samples from the hospitals come to our lab. And we have certain objectives like, understanding what's the burden of disease, what vaccines we need, etc. So because we run the microbiology labs, since the 1990s, when we established the surveillance, we have been trying to keep updated with the diagnostics that are used, the surveillance methods, specifically with a focus on meningitis. So all the CSF samples collected from meningitis patients come to our lab, and in the 1993, we only started with culture. But then we added serology tests. By 2004, we also added antigen tests called ICT. However, in 2002, we received a grant from the Wellcome Trust. And they basically gave us that grant to do a study on etiology and antimicrobial resistance of neonatal sepsis. Using that grant, we bought our first PCR machine, and we got the setup to run PCR reactions. We learned how to design primers, run PCRs, analyze the data, and all the training that we received were generously provided by Professor Derek Crook at the Oxford University. We received the grant to do neonatal sepsis, But we did a lot more than that because we had in-house capacity with empowered local scientists. We used it to detect antibiotic resistance genes in cases of meningitis caused by culture-negative Haemophilus influenzae type B. The data showed us that MDR, Hib, leads to more death and disability. Using that data, we advocated for introduction of the Hib vaccine in Bangladesh, and that was introduced in 2009. The PCR was not funded for this work. We moved on. We also did a lot of work on pneumococcal meningitis. So there are 90 different serotypes of pneumococcus. The vaccine that's available only contains 10 of them or 13. So we had to, and most of our CSF specimens are culture negative. So using PCR, we showed what are the serotypes that are present in the country, and that data facilitated introduction of the pneumococcal vaccine in 2015. Now, we have 4 conventional PCR machines, 2 real-time PCR machines, 1 tilde machine, and we run more than 1,500 qPCRs per month. And this is despite the fact that a fire in 2013 destroyed almost our entire lab. But we got back up because we were empowered. We know how to do science. Using all of that data, in 2016 and 2006, we actually started using conventional PCR to detect etiology in our surveillance platform. And in 2016, we replaced that completely with qPCR. And this is with the help of CDC. We constantly get trained and get refresher trainings from the CDC. Now, despite using all of these techniques to try to find pathogens, in most cases, we don't find pathogen. So in 50% of the confirmed meningitis cases, we are not able to tell what is causing the meningitis. And this is a problem because when you can't tell what pathogen is causing a disease, you don't know how to treat, you don't know how to prevent future cases. So it hinders policy decisions. So I studied in Canada for many years. So when I went back to Bangladesh and decided that my boss will be the person who shares 50% of my genes, I kind of came up with this hypothesis that, even if the culture is set, and it's the same idea of using PCRs, even if our CSF samples are culture negative, whatever organism caused the infection will leave some DNA or RNA. So if we took that entire CSF sample and conducted metagenomics, maybe we will be able to tell what caused the disease. So easy, right? Get a sequencer, do the metagenomics, analyze the data, detect the pathogen. But turns out no one would fund us for a sequencer because there was no evidence that we can sequence. There's no evidence that we can do genomics. And it's not like there is no genomic data from Bangladesh. There's a lot of data, especially look at in the field of typhoid or Salmonella typhi. But the problem historically has been, for all of those publications, a researcher from the global north came to Bangladesh or some other country, Nepal, Pakistan, whatever, took some samples, took them back to their lab, sequenced them, and published them, and got a tenure track position. And here we are, with no capacity, no proof that we can work. So we were stuck in this vicious cycle. No resources to generate data, no data to generate resources, no resources to generate data. How do we break out of that? So early 2018, Farhad Imam visited us, and actually we have been working with the Gates Foundation for a while with this problem, and Karen told Farhad about this problem that we were facing, and we decided that we're going to work with the Chan Zuckerberg Biohub and do metagenomics on some of our samples and see if it can really give us the answer. Now, this is after almost 2.5 years of trying to doing metagenomics with several groups and after several failures. So I was very, very skeptic about the entire process. And before we even embarked on that collaboration, I made 4 points very clear. I said every protocol, every technique that is used has to be open access. At least one local scientist needs to be trained on the method, and all techniques pipelines need to be transferable to our lab and sustainable. And at the end of everything, we will be the owner of our data, and we will decide how to interpret that data. So we embarked on our first such project. It was called Elucidating the Causative Agents of Meningitis Using Metagenomics Phase 1. In this pilot project, we basically shipped 96 samples to Chan Zuckerberg Biohub, and that is where we went through all of these steps. I learned how to extract RNA, how to prepare libraries, sequence, and finally used one of their softwares to analyze the data. Now, I will not go into the details of the results we found, but everything is up on bioRxiv. And what we basically found is using the proper positive and negative samples, we could indeed unravel 50% of those mysterious cases of meningitis. The results were very exciting, and using the data that we obtained, we were actually able to optimize a low-cost diagnostic to look for the pathogens that we found using this very expensive technique. An example of that was chikungunya. So we found that 3 of the samples contained chikungunya virus, but we wanted to test 500 more samples. We could not do metagenomics on all of them, so we just designed a PCR that will specifically look for chikungunya. I came back to Bangladesh, tested 500 CSF samples, and realized that in 2017, Bangladesh was hit by a chikungunya meningitis outbreak that would go completely undocumented had we not gone through this process. So I think that was quite a bit of success for 6 months, and that brought us our second project, which is basically the same project but phase 2. And in this phase, we got our own sequencer. However, the path of getting the sequencer into our lab and setting it up and running the sequences were not easy. There were many, many problems. For example, supply. The reagents, the equipment, it's so hard to get them because these companies like Illumina or QIAGEN or Zymo don't have branches in countries like us. So we have to go through local distributors who can charge us whatever they feel like. However, we got through both the supply and cost chain problems because someone from San Francisco gave Illumina a call, and that is apparently all that's needed to make a difference. And this is after I called them 10 times and sent them many, many emails. Additional infrastructure. So we got a sequencer, but we have not done sequencing before. These machines are very sensitive. They need to be in a room that is temperature controlled, that is humidity controlled. Dhaka is one of the most populated and polluted cities. It needed to be in a room where there's little dust. So we had to basically make a room for it. And all of these problems lead to wastage of time. We spend so much time on it. And for the scientists here who work in labs, it's exciting when you think of an experiment, think of a PCR, order your primers, they arrive in 48 hours and you do your experiment. But for us, we think of an experiment, we order primers, they come 3 months after. By then, there is no motivation to do the experiment anymore, or someone else has already done it. So, these kind of vicious cycles keep us imprisoned in this as scholarly pores. And a lot of scientists who train outside come back to the country, to their native country to work, but then they get frustrated and leave. We got our sequencer and it's running, so none of these challenges are insurmountable. And there are many donors like WHO, CDC, Wellcome Trust, who are helping, but perhaps these efforts can be more coordinated and less bureaucratic. And bureaucracy applies for one of those 3 the most. Okay. So we have used our sequencer several times, And the data that come out of it is definitely extremely exciting. But what makes me most happy is the enthusiasm that I see every time we run that machine. And it doesn't get old. Every time some people in the lab know that we're going to run a metagenomic sample, everyone will huddle in that small room that we have built around the sequencer and try to touch the sample or the machine as if it's their own, and they will wait for the data that will come out after 17 hours. It is just so exciting. Empowering, educating, and engaging local scientists at the front lines of public health can go a long way. So, and why do I say can go a long way? Here are a couple of examples. We got that sequencer to answer the questions of meningitis, but we did so much more. So there is a newspaper, here's a newspaper clipping from mid-August. So 2019 was one of the worst dengue outbreaks in the history of Bangladesh. By the 15th of August, over 50,000 people had already been affected, and just in the 24 hours before that, about 2,000 patients were hospitalized. I work in the largest pediatric hospital of the country. Each day we were seeing 150 patients of dengue. So the question became, why is this outbreak so bad, right? Is it a different strain? Is it a different genotype? What's going on? So we decided this time to sequence dengue. So this is an example of one of the samples that we sequenced. And we were only trained to do CSF, but we figured out we wanted to move on to blood. So this is a case that came to a hospital on the 29th of July. By the 9th of August, we had the sample sequenced and analyzed. But the 16th of August, the sample was already up on NextStrain, and we started our analysis to understand where did the strain come from. We have done more sequencing now, and we know it's not one strain that caused the outbreak. There were several strains circulating at the same time. So we're doing a lot more work on this now. I don't know why things are cut off at the top, but another thing that we have also started doing is, so far we had only been looking at biological specimens, but can we do environmental surveillance to predict outbreaks? So we started we are embarking on a project where we will surveil mosquitoes to understand what are the viruses in them. And this is actually extremely exciting for our lab. So the mosquito that you see is an Aedes aegypti. This mosquito was actually captured in our lab right beside our PCR hood. And the student who captured it is very excited to know if there is dengue in there. And we are also using metagenomics. We just got funded for this project, actually, to understand how Salmonella typhi survives in water. So those are just some examples of how a local capacity building can go very far. As a group over here of the human cell analysts, if we really want to make changes, if we really want to build capacity in the local level, I think we have to come in terms with the fact right now that the path forward is not going to be easy. The path forward is going to be a path of resistance. It is going to be a path of a lot of hurdles. Building infrastructure from scratch and suddenly is very hard and probably not sustainable, and this has come up in the previous talks too. How do we find existing infrastructure? How do we find the labs that already work well? For example, the labs funded by WHO, CDC, or the Wellcome Trust labs around the world. And also, just because a lab has a lot of equipment doesn't mean the lab works very well. So in Bangladesh, there are at least 15 Illumina sequencers. If you do a PubMed search, I don't know how many pathogen sequences you will find. We got our sequencer just a few months ago, and every time we sequence, our local distributor pays us a visit, and he told us that this is the most anyone has sequenced. So planning ahead is going to be key. We need to figure out if we are going to embark on this project, we need to figure out if we are going to do the experiments, plan the study, where will the reagents come from? Even if the reagents need to be taken in a suitcase, it needs to be planned ahead. What will the publication be like? What is the roadmap to the success of the study? And of course, as we talked about before, how do you define success? The path forward will require dedication, effort, patience, and trust. And failure is not an option. And let me make this very clear. If we really embark on this project, and if we fail, what's going to happen is the PIs from North America, nothing is going to happen. They're going to go back to their labs. They're going to continue with their NIH funding or whatever. But all the donors will remember that an attempt was made to build capacity, and it failed. And every single time in the future, this example is going to be used. You know, we tried, we failed. What's the point of trying again? We cannot afford to fail. I'm going to end with this slide, that this is our opportunity to empower, engage, and educate. This is our opportunity to intercept the colonial world of genomics. I've tried to avoid the word colonial throughout my talk, but here it is. And this is our opportunity to ensure everyone across the world has equal access to the practice and benefits of science, because science should be by and for the many, not the few. Thank you.
Thank you very much. No burning questions? Shyam.
Senjuti, I hope I got that right. Brilliant story. And just one quick comment. There are people doing microbial surveillance for pathogens even in Singapore, I know for a fact, and others, Southeast Asian countries. So it would be fantastic to develop connections in the area. So looking forward to that.
Sure.